So this is something of a challenge, but they were kind of designed for good reasons. But we also have tried to make this data as accessible as possible to as many people as possible. People who are just interested in a single gene and are not interested in 90 terabytes of data. So we built a browser for the data. It's built on the Ensemble platform. We update this about every 2 months as more data comes out of the project. And so this is just an example of what you see now with the pilot project data. So and by the way, I should mention all the pilot project data was released before publication back in March. And so it's been available for anyone to use in advance of the publication, all the SNP sets and things like that. So here you can see the SNPs associated with the different pilots and the different populations. If you click on a given link, you can export data from the window. And in fact, for a few genomes, we also have the ability to go in and look at all the aligned sequencing reads. This is currently just available for the high coverage trios, and we're trying to figure out how to engineer to do this for most of the data sets that we have. Going in and looking at all the aligned sequencing reads is a relatively specialized activity, but some people do want to do it, and again, without downloading 90 terabytes of data to do this themselves. So we've also been experimenting with other ways that we can make these data sets available as widely as possible. And one way of doing this is working with Amazon. The Amazon Web Services people have essentially agreed to host the 1000 Genome Pilot Project alignment files in the cloud as a public data set. So this is about 10 terabytes worth of data that's accessible by the Amazon cloud. Anyone can go and use this for as little as 8.5 cents per CPU hour, which is actually really interesting. As it turns out, not a lot of people, to my knowledge, have accessed these data sets. But it is available, and people like Lincoln Stein, as well as Steve Salzberg's group at the University of Maryland, have not only called for a greater use of cloud computing and genome informatics, but also, in the case of Steve's group, built a number of tools that are designed to use cloud infrastructure to do genome analysis. So it will be interesting to see how this goes. We do have other experience with using biological data sets in the cloud. So one of the public data sets on Amazon Web Services is the Ensemble data sets. Besides being available as a backend for our genome browser, we have them available within the Amazon cloud, and anyone can access them from there. They're actually the second most popular data set in Amazon's public data sets, and so we know they're used, and they're used reasonably widely, including by us, actually. We use the Amazon public data sets as a backend for a mirror of the Ensemble genome browser that we deployed on the US East Coast. And so this turns out to be a valuable way for us to distribute data to more people than they would otherwise get their hands on it. So the first challenge is about creating and managing the variant data. But once we have these SNP sets, so we've got 15 million SNPs in the 1000 Genomes Project. So what? What do these things mean? Do they mean anything? They might mean very little. It might be just a class of normal variation. But the goal is, I think, to try to find this out as quickly as we can. So how does one go about finding which variants are important? So one way is to look in protein coding or other known genes. So this is actually relatively easy. Almost all of the locations of the protein coding genes are known in the human genome. Of course, the full extent of polymorphic pseudogenes is simply not known. We haven't taken enough time to study it, and we haven't had enough samples to know how often a given gene is just disabled in the fraction. And so as we do more samples and we get a feel for the extent of polymorphic pseudogenes, this will tell us a lot about genome biology. And so whether or not you see a stop codon in a given gene, it may turn out that 40 or 50% of the population has a stop codon in that gene, and so you can just ignore it in your genetic studies. So another way to find out which variants are important is to look in conserved non-coding sequences. This turns out to be an imperfect filter, though. We know from work done in my group, as well as a lot of other work in functional projects, that the actually used transcription factor binding sites in regulatory regions change very rapidly. And in fact, as far as we can tell, functional transcription factor binding sites don't exhibit higher sequence constraint than those that are apparently unfunctional or non-functional. Another way to figure out which variants are important, and I think this way is going to be increasingly popular over time, is to integrate functional annotation and other experimental results and to overlay these with the variant maps that we're developing. So one of the ways that we integrate this information, we think it makes sense in that we have a platform for data integration already, and a way that people are used to approaching it is through Ensemble. Most of you have probably heard of or used Ensemble, but our goal is to create and enable genomic science by providing high quality and consistent annotation in a way that it's easy to access and available in a number of different core data sets, such as our gene sets, multiple alignments, and other pieces of information. And so we'd like to make things easy to access through the system that we already have. I'll give you a couple examples of that. So Jeff has already talked about genome-wide association studies. There's a lot of genome-wide association studies that have come out. Unfortunately, databases that just simply store these and map these are actually not very useful at present. There's a number of phenotypes that exist. We have something like 60,000 phenotype annotations, which is not to say that there's 60,000 SNPs that have a phenotype annotation. But unfortunately, if you go to something like the NHGRI's GWAS catalog, which they've done a good job of collecting these data, they don't store disease information using an ontology. So diabetes with a capital D and diabetes with a lowercase d are two different diseases. And this makes it difficult for one to go through and coordinate these data sets. So we've been working with this, and in fact with NHGRI, to try to make these resources more useful. We've also been working with groups of LSDBs. These are locus-specific databases. Some of these are very valuable, and they're at the end of diagnostic labs pipelines or other clinical resources to get these data shared into wider use and into to common integrated resources. Earlier this year, working with partners at NCBI, we published a standard to do just this, which we call the LRG standard. Nature Genetics, in the following month's issue, wrote an editorial in support of using this standard as a way to report genetic variation in a consistent way that we can use all the way from the genome browsers down to the clinic. So here's an example, actually, of one of these things as it really looks in Ensemble. So here you see Ensemble genes, and these purple areas correspond to the LRG sequences. These are really a way for information to be transmitted between groups that it was difficult for information to be transmitted between before, between a diagnostic lab and a genome browser. And we're trying to break down those things that prevented information to transfer between these two, to make the data that comes out of diagnostic labs more accessible, and to make the information that's collecting on the genome index in something like a genome browser more immediate to clinicians who might want to use that information. So we've also built some tools to do automatic annotation of discovered variants across the genome. So the first of these is called our SNP effect prediction tool. We're working on the additions to this to make it general for any structural or other complex variant. That turns out to be quite difficult. But what it does now is it calculates the effect of SNPs in the context of all the Ensemble genes and the regulatory features. So this will tell you immediately on any given SNP in any species that Ensemble supports, whether or not it disrupts the protein coding sequence, it disrupts a splice site, it's in the 5' UTR, all of these type of annotations. We work directly off of VCF files or website upload. And we're currently working in the context of the ICGC, the International Cancer Genome Consortium, to capture structural and these other genome rearrangements, which actually come in a mind-boggling array of different constructs and flavors. In early 2011, we're going to take the SNP effect predictor up into the annotations produced by the ENCODE project. And so if there's a transcription factor binding site motif that's been identified in the ENCODE project, and a SNP hits that motif, we'll be able to return that result basically as fast as we can do the lookup. So we have a relatively simple web form for this, which is limited for a number of reasons to several hundred SNPs, or about 750. It works, data submitted, results are returned, including RS numbers, if it exists in dbSNP, as well as, you can see the annotations here. This results in a stop codon being disrupted. These are associated with a frame shift coding and things like that. So we think this is a valuable tool for automatic annotation of many variant pipelines. And it's non-trivial to do this by just simply downloading a bunch of files from a genome browser and writing code to do yourself. So we want to continue to take this forward as we develop more and more annotations in the genome, more that could be affected by SNPs, so at least you know when a SNP hits something that's already been annotated. This is an example of differentially methylated regions in the genome. These are things that we hope to return with the SNP effect predictor. And going forward, as we create integrated regulatory annotation based on data sets from ENCODE and the Human Epigenome Roadmap project, this is just an example of the way we're pulling in all of the data sets that have come out of embargo in these projects, and so can be incorporated into Ensembl through a consistent set of alignments and then peak calls, and eventually they end up in little multi-wiggle tracks on our browser, and I'll show you an example of that here in just a second. So this is an example of a regulatory region around, I think this is the RPS12 gene, and this regulatory feature, as we call it in Ensembl, is built on a whole host of data generated from both the ENCODE and Epigenome Roadmap projects. And you can see if we zoom in here, how these different tracks overlaid each other and where we see peaks. And as we go forward, we want to develop increasingly sophisticated statistical models that take in an input like this and tells you what sort of regulation is going on. Other people are working on these same projects, and I think that the rich data sets in ENCODE are very valuable to do this. So this is just an example of how we're now painting regulatory features across the genome. Almost all of this data, as I said, is coming from the ENCODE project or the Human Epigenome Roadmap. As the International Human Epigenome Consortium continues to go forward, we anticipate a whole host of this data becoming available in all sorts of different cell types and lineages. So finally, I want to talk a little bit about how to create an infrastructure to find and annotate variant data as quickly as possible. So the basic question here is, has this variant ever been seen before? People are doing all sorts of variant discovery, from whole genome sequencing to exome studies. And at the end of the day, if you read their methods pipelines, it's a bioinformatics jujitsu nightmare of collecting all of these different variation data sets, trying to filter your set against them, just to answer this simple question: has this variant ever been seen before? Should we care about it? In an exome study, you're going to find 20,000 variants, and to filter them down to the 1 or 2 or 10 that might be important is I think, a key question, and one that we're going to need to answer if we want to transition genome sequencing into a viable clinical screening tool, which it's potential that it could get to. So if you look in Nature's issue today, Nature says that about 2,700 genomes have been sequenced. They estimate that 30,000 will be done by 2011. But beyond those that are in the 1,000 Genomes Project, which is around 1,100, very few of these genomes are actually easy to access. They exist in disease studies. They're Ozzy Osbourne's genome or Glenn Close's genome. And you just can't kind of go out and ask, has this variant ever been seen before in these genomes? This is equally true of many of the exomes. Some of the data is available, but it's not easy to access and answer this relatively simple question. Some of it is not even freely available, and shouldn't be actually, because it's under controlled access or managed access because of the consent agreements that the research participants have signed, and this category is expected to increase.